So enrolling into a gene therapy trial has similarities to other clinical trials that people may have been involved in, whether that be extended half-life or other factor trials. That would be about a conversation about whether they're eligible, reading the patient information sheet to ensure that they had the full background of the trial, and having ample time to ask questions of the clinical trial team. Ultimately, for any clinical trial, a patient will need to sign a consent form to say that they understand the trial structure, the requirements and the risk. And that's where the gene therapy side of a clinical trial is different to some of the other trials, because you end up consenting into a trial that's quite unique. Medicine, when it's infused into you, can't be taken out again. So it's very different to an extended half-life that once one's been given, it will fade away very quickly before the next infusion of EHL is given, whereas with gene therapy, it lasts forever. So it's very different in thinking about consenting to the trial. And this is really, for me as a clinician, the most important thing that an individual understands, that once it's in, there's no way of neutralizing or putting it to an end once it's in your body. So this is where it's so different to other trials that you can't opt out of the gene therapy. So this is something that makes the clinicians nervous, and I would imagine makes individuals considering the trials nervous as well. It's impressive to see the number of men who've stepped forward, who've wanted to participate in the trials to date, both in the UK and elsewhere in the world, who have been happy to take this risk on. And fortunately to date, the trials have been progressing well, but it's an absolutely important message that once it's in, it can't be retrieved and taken out again. So ahead of the infusion of the gene therapy, there are no particular special requirements. There's no requirement of liver biopsy, there's no additional invasive procedures. The infusion of the gene therapy itself is very simple via a vein in the arm, which is infused during the day, and generally people would be able to be discharged the same day, or certainly after a single overnight stay. So I think the actual logistics of once consented and then having gene therapy delivered is pretty simple. After the gene therapy has actually been administered, the team will watch the expression of the given factor very closely. There'll be multiple time points in a given week and over the first months to watch the factor as to how it's expressed. Depending on the different trials, there may be subtle differences as to how quickly the factor is expressed, but it's certainly weeks and months, not days and weeks. Thinking about the factor expression level after gene therapy is an important consideration when you're thinking about participating in a clinical trial. The different platforms, so the different companies bringing trials to your centre may have different targets, they may have different doses, and even for the same company there may be a choice between a high dose and an intermediate dose or lower dose. So this is an important conversation to have and to ask your clinical team and the trials team what the different platforms mean by a high dose or low dose, and often to ask how they compare to a different trial, maybe even if that trial is not open in that centre. So the difference between gene therapy trials and some of the other factor trials is that often a centre may only open one trial for gene therapy rather than multiple. But just because that trial is open does not necessarily mean that's the right trial for you at your centre. So I think it's important to feel confident to ask questions, not just about the trial that is open at your centre, but how that trial compares to other trials available in your country. So after gene therapy, there is a risk of a reduction in the expressed factor level. And in some trials, this has happened quite soon after the infusion. This may be related to some liver function tests becoming elevated and sometimes requiring steroids to treat. It also seems from published data that factor levels in other trials settle after a longer period of time, maybe after a couple of years, to lower levels than was seen in the early months after gene therapy. The question then is whether this matters and whether the settled level is still sufficient to protect the individual from bleeds and whether it's really gone back to a proper severe haemophilia or whether it's left the individual with mild haemophilia that may still be very protective against bleeds. So this will need to be considered on a case-to-case basis, but if the factor level went all the way back to severe form, the unfortunate situation is that you cannot be redosed with the same gene therapy, and it may be the case that you can't be redosed with other AAV gene therapies either. But this is again an area of research, and we're uncertain about the cross-reactivity of antibodies against the different types of adeno-associated viral vectors. So for those that have levels that are still moderate or mild haemophilia levels, there will be times that they need additional factor top-ups, and this theoretically could be a standard half-life or an extended half-life product, and we would have no concerns about that. The requirements after the gene therapy has actually been infused is quite time-consuming for the individual who's received the gene therapy. So depending on the trial, there will be multiple visits to the haemophilia centre, the trial centre that's overseeing all the sample collection, and that's going to be multiple blood samples, and that in the first weeks and months will sometimes be more than once a week. And then also collection of bodily fluids, so that would be saliva, urine, semen and stool would be collected. And this is an important part of the trial that people need to be aware of as to whether they're prepared to do that. But it's scientifically important, and that's the reason we need to understand how the virus escapes the body, its so-called viral shedding, and how long it survives in the body and how it escapes from the body in the different body fluids. The individual undertaking gene therapy will need to listen carefully and discuss how the gene therapy trial might impact on their personal relations with their partner, and certainly the conversation about contraception and the requirement for contraception over a prolonged period of time. So certainly anyone considering starting a family in the near future, it's an important consideration as to whether the time is right for them. And the reason why contraception would be important would be because of the risk of the virus being shed in the semen of the man and the unknown risk to their partner if they were trying to get pregnant. So for the guys with female partners that are thinking about families, this is a very important consideration. So the eligibility criteria for enrolling into gene therapy trials have similarities to some of the factor trials of years past. So currently it is adults for gene therapy. There's no pediatric studies yet. The main issue will be around liver health as well. So the individuals need to be free of hepatitis C in a sustained virological response from their hepatitis C treatment, and that their liver is in good health as judged by a fibroscan normally, so a scan that is not needing an actual biopsy of the liver, but many individuals will have had these scans done to assess their liver, and that would certainly be done to assess liver health. There'd be the usual blood tests to assess liver health as well. Currently, different trials may have different criteria around HIV, and in some trials that's a pause exclusion at the moment, and there's thought about whether this would be a true exclusion or not. A key thought is about inhibitor status. So similar to the factor trials of times past, without an inhibitor history and with very clear blood tests and recent blood tests, one of which that will have been done in the central laboratory to demonstrate that there's no current inhibitor. The long-term exclusion in the real world, if and when gene therapy gets to market, we obviously will need to understand how these groups that are currently excluded may or may not get access to gene therapy. So currently, the group of women who may have severe haemophilia is a very, very small group and are currently not eligible for trial. There are clearly women who live with haemophilia, but that tends to be mild or moderate haemophilia, and that's an exclusion to current gene therapy trials. The challenge in the field at the moment is thinking about rare disorders beyond haemophilia A and B, but currently the only gene therapy trials are for haemophilia A and B. So although there is a need in the wider community for thought about gene therapy, there are not the clinical trials open or imminently coming to the centres for trial participants with rare disorders. So I get asked in clinic quite a lot about mild and moderate haemophilia as to whether they would be eligible for gene therapy. And disappointingly for them at the moment, they're not eligible. But I think it raises an interesting question if gene therapy is available to the most severe individuals, those then with the variation of moderate haemophilia A or B clearly will start to think that they should be eligible, and I think this will be an ongoing conversation. Currently they're not eligible for gene therapy. So currently the age criteria for enrolment in gene therapy trials is for adults only, and understandably there is caution within the community about progressing into adolescence and certainly to younger children, including adults only. Theoretically, the best people to treat would be the youngest patients with gene therapy. So philosophically, the earlier the gene can be corrected or replaced, then that would give advantage to the young boy with potential severe haemophilia. So I think we need to continue to watch the adult progress very carefully, and clearly from the original UCL St Jude's trial back at nearly 10 years ago, and then the more recent trials, we have a good number of adults who have had years of experience now with gene therapy. So I think there's an ongoing conversation about when younger adults, adolescents, and then children might be eligible. So in my opinion, gene therapy emerging as a new therapy for haemophilia brings great opportunity for the community. This comes at the same time as other novel agents come into market, other modified factor 8s and factor 9s, as well as our standard half-life factors that we've used for so many years with great safety and great effect. So in my view, all of this landscape change brings opportunity, but I don't think there's going to be one treatment that becomes so dominant that the other treatments will disappear. Individuals and their families will need to decide which factor suits them best for their haemophilia, and certainly already within departments we're seeing the variety of opinion about gene therapy, that some are super keen to get involved, others are interested but happy for others to proceed, and others are very happy to hear about it but have no intention of actually receiving it. So I think this is likely to reflect what happens in the next decade as all of these new therapies, including gene therapy, progress, that there will be a spectrum of enthusiasm both from the individuals, families and the treating teams in hospitals as to what treatment is right for the right patient at the right time.